Gaudeamus igitur, iudenes dum sumus. Gaudeamus igitur, iudenes dum sumus. Post iucundum juventutem, post molestam senectutem, nos
Good evening, friends and colleagues. It gives me great pleasure to welcome you this evening at the inaugural address of Professor Gyebi Duodu from our Department of Consumer and Food Sciences in the Faculty of Natural and Agricultural Sciences. I would now like to introduce the Vice-Chancellor, Professor Tawana Kupe, to provide a brief biographic background to Professor Duodu. Professor Kupe.
Good evening, ladies and gentlemen. Thank you, Mr. Dean, Professor Barend Erasmus. Thank you, Professor Duodu, the inaugurating professor. Professor Duodu is Professor of Food Science in the Department of Consumer and Food Sciences, University of Pretoria. His association with the University of Pretoria goes back a little more than 2.5 decades, when he studied for a master's degree in food processing in the Department of Food Science after obtaining a BSc degree in chemistry from the University of Ghana. Prof. Duodu went on to obtain a PhD in food science from the University of Pretoria in 2000. After a brief stint as a lecturer at the Tshwane University of Technology, then Technikon Pretoria, Professor Duodu joined the University of Pretoria in March 2002. He was promoted to senior lecturer in January 2006, associate professor in January 2013, and full professor in January 2020. When he joined the University of Pretoria, Prof. Duodu established an area of research focusing on the health-promoting properties of African grains and their foods, which we are going to hear more about this evening. In his research, Professor Duodu investigates the metabolomic, profiling, chemistry, bioactivity, and health-promoting properties of the phenolic compounds, specifically concerning diet-related non-communicable diseases. Professor Duodu is a member of the Bioactive Compounds Committee of the Cereals and Grains Association, formerly American Association of Cereal Chemists International, and the editorial boards of the Journal of Food Composition and Analysis, Legume Science, and ASC Food Science and Technology. He has been either the primary author or co-author of more than 60 articles in international peer-reviewed journals, 12 book chapters, and co-editor of the recently published Sorghum and Millets, Chemistry, Technology, and Nutritional Attributes, AACC International. Professor Duodu has contributed significantly to capacity building in food science and technology research on the African continent, having trained, mentored, and supervised 18 doctoral and 29 master's graduates from various African countries. Outside academia, Professor Duodu is a musician, and he likes to describe himself as a budding pianist. He's convinced that he would have surpassed his achievements as a scientist by choosing music as a career. But for today, he will speak as a scientist. Prof. Kwaku, Gyebi, Duodu, your audience tonight.
Professor Tawana Kupe, Vice-Chancellor of the University of Pretoria, Professor Barend Erasmus, Dean of the Faculty of Natural and Agricultural Sciences, University of Pretoria, Professor Elna Bays, Head of the Department of Consumer and Food Sciences, University of Pretoria, colleagues, family and friends, good evening. I'm extremely grateful for this opportunity to present this inaugural lecture. And I'm also grateful to you for your interest and taking the time to listen to this lecture. It is on a topic that is very dear to me and has occupied my research endeavors for close to 2 decades now. So as you can see, the topic of the lecture is phenolics in African grains and their foods, a tale of their significance for health and food security. So I intend to speak to you about these compounds known as phenolics and their significance for health promotion. But more importantly, I would like you to take home with you a keen awareness of what we are referring to here as African grains and their importance for health and food security. Perhaps a logical place to begin is to get an understanding of what phenolic compounds are. Now, phenolic compounds are actually a very large class of plant secondary metabolites, and they show great diversity in terms of amount and type. So in other words, it simply means there are many of them, and there are many different types of them as well. Now, where our African grains of interest are concerned, we conveniently divide phenolic compounds into 3 main classes, and this is in order of increasing molecular weight: phenolic acids, flavonoids, and condensed tannins. Now, strictly speaking, because they are simply polymeric forms of flavonoids, the condensed tannins are sometimes simply classified as flavonoids. However, due to the fact that they exert some important nutritional and sensory effects, it helps to study condensed tannins as a class on their own. And I'll be talking a bit more about condensed tannins in this lecture. So why are phenolic compounds important? Well, for us, the answer is simple. Phenolic compounds are important because they exert potential health-promoting effects, particularly in regard to diet-related non-communicable diseases. Now there is this quote, "Let food be thy medicine, and medicine be thy food." It's attributed to Hippocrates, the ancient Greek physician regarded as the father of medicine and for whom the Hippocratic Oath is named. But for us, what this quote brings to the fore is the thinking that food should be viewed as not only providing nutritional benefits, but also health benefits. Therefore, for us, the link between food and health is quite important, and it has become an area of great interest, and we believe that phenolic compounds play a significant role in this regard. Now, in order to better understand the evolution and metamorphosis of the field of dietary phenolics and health, a brief historical perspective would be useful. Now, the compound phenol itself is said to have been discovered in 1834. But for the purposes of this brief historical review, I would like us to begin from the 1940s. And let's look at the period from the 1940s to the 1960s. Now, this period featured some really early papers that were reporting on antioxidant properties of phenolic compounds in oils. One example is a paper in the journal Oil and Soap by Golombek and Mateo, which reported on the antioxidant properties of garlic acid in vegetable oils and animal fats, and this was in 1942. Now, within this period, you begin to see studies reporting on tea catechins, perhaps not surprisingly. And we're looking here, for instance, at work done to analyze tea catechins using paper chromatography, and those were predominant during this period. And that is quite interesting when you think about it. When you consider that these catechins at that time were being characterized using paper chromatography, and you compare that with the current state-of-the-art analytical techniques we use, such as liquid chromatography, mass spectrometry, it gives you an indication of how far things have advanced in the field. But then the study of phenolics during this period was by no means restricted to catechins in tea. There were also reported studies on anthocyanin pigments and tannins in cocoa, phenolics in brewing materials, studies on glycoflavones in wheat and oats, and proanthocyanidins in apple and grape. Now we're talking about phenolics in health. Interestingly, one of the earliest reports on health promoting properties of phenolics was actually an in vivo study of the influence of dietary phenolics on, of all things, frostbite in the rat. And this was in 1955 by Frederick Furman. And this was published in the American Journal of Physiology. And then in 1962, there was a study on antichoriogenic activity of phenolic acids in oat hulls, reported by Vogel and co-workers in the Journal of Dental Research. Now, something else which is quite important. In this field of phenolic antioxidant research, structure antioxidant activity relationships are quite important. Now, what do we mean by that? It simply refers to the thesis that the antioxidant activity of a phenolic compound is related to its structure. And so in this regard, one of the earliest reports on structure-activity relationships of phenolics was by Miller and Quickenbush in 1957 in the Journal of the American Oil Chemists Society. And that was quite interesting. These authors showed that a free phenolic hydroxyl group and also occurrence of electron withdrawing substituents on the phenolic ring were important for antioxidant activity. Now let us move on to the 1970s and 1980s. Now during this period, there were more reports on phenolics, this time in grains. So grains began to feature a bit more. So we're talking about, there were reports on phenolics in things like cashew, oats, rapeseed, sorghum, and beans. And now talking about grains, sorghum is actually of particular interest to me, as a lot of my research on phenolics, as you will see later, has been on sorghum and sorghum-based foods. And so in this regard, one of the earliest reports about studies on phenolics in sorghum, which caught my attention, was one on the relationship between tannin content, enzyme inhibition, rat performance, and characteristics of sorghum grain by Maxson, Rooney, and Lewis. And this was in 1973. Now, this caught my attention. It's quite important to me because one of the co-authors of this paper, the late Professor Lloyd Rooney, was actually known to me personally. And he was a colossus of sorghum and millet science, and all of us in the field have benefited tremendously from his trailblazing and pioneering work. I also need to mention early contributions of South African scientists to the study of polyphenols, in sorghum specifically. An example of this is a published study on enzyme inhibition by polyphenols of sorghum grain and malt by Klaus Daiber of the CSIR, and this was in 1975. Now, in this field, a significant contribution to the study of phenolics in sorghum in particular was made in 1978 by Gupta and Haslam, and they published an extensive chemical characterization of sorghum condensed tannins. I'll be talking about sorghum condensed tannins in this lecture, and they showed that the structure of sorghum condensed tannins had a nature where it consists of catechinic terminal and extension units as well. And then coming to health promoting properties, there was a study on anti-mutagenic activity of green tea polyphenols, and this was in 1989 by Wang and co-workers in the journal Mutation Research. And then this was followed by a similar study on anti-mutagenicity of polyphenol-rich fractions of sorghum grain in 1992 by Grimmer and co-workers of Wits University. So that was quite interesting. But then in general, in the 1990s and 2000s, that period was characterized by an explosion of research reports on health-promoting properties of phenolic compounds, specifically in relation to diet-related non-communicable diseases. And importantly, these studies highlighted structure-activity relationships. So in this regard, a review by Rice Evans and co-workers on structure antioxidant activity relationships of flavonoids and phenolic acids, which was published in Free Radical Biology and Medicine in 1996, was quite pivotal. And then during this period as well, bioavailability of phenolics began to grow in importance as a research focus. There was a report on plasma and tissue levels of T catechins in rats and mice during chronic consumption of green tea polyphenols. And this was in 2000 and published in the journal Nutrition and Cancer by Kim and co-workers. So from this brief historical perspective, it is clear then that by the 2000s, phenolic compounds in foods and their health-promoting properties had evolved and grown to become an area of intense research interest. And it is at this time that I enter the timeline of the dietary phenolics and health story. And when I joined the University of Pretoria as a full-time academic staff in March 2002, my mentors at that time, Professor John Taylor and the late Professor Amanda Mena, stressed the importance of working towards standing on my own as a researcher. In simple terms, they advised that I needed to identify, establish, and grow my own unique research area. And my response to this was a decision then to focus on the field of dietary phenolics and health. Now, given my background in chemistry, the choice to focus on this research field was actually quite easy, as it offers the opportunity to engage in some exciting and interesting and quite stimulating chemistry. However, more importantly, my choice of this field of research is born out of a strong conviction that it has the potential to impact positively on people's lives. Allow me to provide some context here. In Africa, reference is made to what is called the triple burden of malnutrition. What do we mean by that? Well, there are three legs. One of them is micronutrient malnutrition, and macronutrient malnutrition. Those are well known in Africa. But then a third form of malnutrition, namely overnutrition, has emerged in Africa in recent years. Rapid urbanization, rising incomes, and the quest for convenience has led to poor dietary choices and produced what they call a nutrition transition. So this, coupled with relatively low levels of physical activity, has driven the occurrence of obesity, metabolic syndrome, and in general, diet-related non-communicable diseases in Africa. I would like to take South Africa as an example. Data from the World Health Organization indicates that there has been a steady increase in the proportion of deaths due to non-communicable diseases over the years. Starting from 2011, the proportion of deaths due to non-communicable diseases in South Africa was around about 29%. Move on to 2014, and that had jumped up to 43%. And then in 2018, went up further to 51%. So that's quite alarming. I do not have the data for 2019. It will be interesting to see how the COVID-19 pandemic is going to influence the figures for 2020. But with this background in mind, the thesis on which my research area is based is that dietary phenolics can play a role in modulating physiological processes that are brought on by overnutrition, and by so doing, potentially offer protection against the occurrence of and mortality from diet-related non-communicable diseases. And so, as important sources of these phenolic compounds, we believe that African grains can play a significant role. So the logical question to ask then here is, why African grains? What's so special about them? Okay, just about over a week ago, the Intergovernmental Panel on Climate Change presented its 6th assessment report on climate change. And unfortunately, I'm sure you'll agree with me, it was not good news. Actually, the UN Secretary General, Mr. Antonio Guterres, described the report as code red for humanity. Quite alarming. In terms of agricultural productivity, most of Africa will be negatively affected by climate change, as shown in this map, which was produced by the European Environmental Agency. So this has some implications. We believe that in this climate change era, crops that will be important food sources for Africa should be drought tolerant. They should have a generally low water footprint. And in addition, they should be nutritious and health promoting. And we are proposing that African cereal and legume grains are the perfect candidates. So I'm giving examples of cereals such as sorghum and millets. And over here, I'd like to thank my good friend and collaborator, Dr. Saikat Mazumda, for this slide, which shows the sorghum and millets quite nicely. And then also, we're interested in climate-smart African legumes. We've done work on cowpeas, we've done work on Mambara groundnuts, and then we've done work on perhaps maybe the lesser-known Marama bean, which I'll be talking about a bit later on. Now, most of these grains are believed to have African origins and are also relatively drought tolerant, and therefore, and they have a very wide variety of food uses. So we've got these grains. So the question is, how are they used for foods? How are they utilized? So the utilization of African cereal and legume grains, I like to classify them into 3 main levels, very broadly. Utilization level 1, well, this level involves the use of what I'm referring to as simple, traditional processing methods, which may be loosely described as low-level technology. So we're thinking about processes such as cooking, simple cooking, boiling, fermentation, malting, roasting, etc. And these will normally be used to produce mainly traditional foods at the household level. And then utilization level 2, this level involves the use of modern high-level technology processing methods like extrusion, micronization, microwave processing, etc. An example would be the use of such technologies to produce ready-to-eat foods that may be targeted towards the urbanized consumer. And then utilization level 3 is what I refer to as waste reutilization. And this is simply where byproducts from primary processing of cereal and legume grains are reutilized. Example, using bran and seed coats as sources of bioactive phenolic antioxidants for various food applications. So what I would like to do now is to share some of the research we have done to show the health promoting properties of phenolics in African cereal and legume grains at these 3 levels of utilization. We're going to begin with utilization level 1. And I'm going to talk a bit about some of the work we've done on health-promoting properties of sorghum cowpea composite porridge. Now, this work was done by Dr. Franklin Appiah-Ban, who did his PhD with me, and where he was looking at a very simple food system, a composite of sorghum and cowpea, and we're trying to find out its health-promoting properties in relation to phenolics. Now, normally, cereals and legumes are combined in composite foods to achieve enhanced protein quality. Why is that? Cereals are generally deficient in the essential amino acid lysine, but contain adequate amounts of sulfur-containing amino acids. On the other hand, legumes tend to contain relatively low levels of sulfur-containing amino acids, but are rich sources of lysine. Therefore, the cereal legume composite food will contain a better complement of essential amino acids for enhanced protein quality. So in Franklin's work, we saw an opportunity to combine sorghum and cowpea in a composite porridge to achieve enhanced phenolic profile and health promoting properties. And why do we say that? This is because when we characterize the flavonoid components of these two grains using LCMS, we found that whilst both contained flavanthriols and flavonones, sorghum contained flavones which were quite absent or in low, very low levels in cowpea, and the cowpeas in turn contained flavonols which tend to be absent in sorghum. So the opportunity here we saw that the sorghum cowpea composite would contain a more complete flavonoid profile with contribution of flavonoid compounds from each flavonoid class. And that's exactly what we found, as you can see in this table. But going on further from that, we hypothesized that the composite porridge would have enhanced antioxidant properties if we took structure-antioxidant activity relationships into consideration. And some structural features of flavonoids that are important for antioxidant activity are the following: a 3,4 catechol group on the B ring, a 4-oxo functional group adjacent to and in conjugation with a C2C3 double bond in the C ring, and OH groups on carbon 5 and carbon 7 of the A ring. So you see that the presence of the 4 groups of flavonoids in the sorghum cowpea composite would bring together the best assembly, I'll call it, of all these required structural features for enhanced activity, antioxidant activity of the composite porridge. And that is what we found. We found additive effects of radical scavenging properties in the composite flour. So if you look at this table, we're looking at the interactive effects on radical scavenging properties on compositing sorghum and cowpea flours. And we're looking in particular at the sorghum cowpea flour column, and we're looking at how close our observed value gets to the expected value, which was calculated using principles of mass balance. And then we also moved on to try and determine health-promoting properties of these sorghum cowpea composite porridges. And as you will see in this slide, they exhibited ability to protect membranes of erythrocytes from oxidation. Now, the second system I would like to talk about is work we did on cowpeas. So this is just about cooked cowpeas. And Dr. Alice Inderitu and Dr. Twambo Hachibamba did their PhD with me, and they conducted their doctoral research on phenolics and health-promoting properties of cowpeas. And this was actually part of a USAID Dry Grains and Pulses Collaborative Research Support Program project, which was on increasing utilization of cowpeas to promote health and food security in Africa, where we collaborated with Texas A&M University, University of Zambia, and Egerton University in Kenya. The main objective, what we were interested in here, was to determine the effect of cooking and simulated in vitro gastrointestinal digestion on cowpea phenolics and their health-promoting properties. And of course, we used techniques like UPLC to determine the phenolic profile, and looking at the phenolic profile, various phenolic acids and flavonoids from the flavanthriol, flavonone, and flavonol classes were identified in the cowpeas. And then we went on to try and look at health-promoting properties. And one of the ways we determine that is to look at gene expression for what we call here TNF-alpha. TNF-alpha simply stands for tumor necrosis factor alpha. That is a cell signaling protein or kind, and it is involved in systemic inflammation. So it is used as a biomarker for inflammation. And the work showed that these cowpeas showed ability to reduce gene expression for TNF alpha, as you can see in these figures. We're also interested in DNA damage, one of the ways we determine health promoting properties. And if you look at the DNA, it originally occurs in a supercoiled form. Now, when it gets damaged by oxidation, it assumes other forms, like an open circular form, a linear form, and when the oxidation is quite extensive, it gets fragmented. Now, all these forms of DNA have different levels of mobility in a gel under electrophoresis, and that's oxidative damage. And one final one is looking at gene expression for phase 2 detoxification enzymes. A typical example is quinone reductase over here. So it's a phase 2 enzyme, and it provides some protection by detoxifying the body by removing carcinogenic compounds from the body. And so gene expression for quinone reductase is an important way by which we can determine health promoting properties. And we found that raw and cooked cowpeas actually showed significant induction of the activity of quinone reductase. Now, I cannot talk about African foods without talking about soured foods, because soured grain-based foods are actually quite important in Africa. They are consumed extensively in many African communities. In general, souring can be achieved using 3 main methods. We can use spontaneous fermentation, which is actually practiced in many African households, or by using specific lactic acid bacteria starter cultures, and this can be done in the manufacture of products such as maheu. And then exogenous acidification to achieve souring is also widely practiced at home level, where fruit acids or juice from citrus fruits may be used to sour cereal foods instead of using the longer and maybe more cumbersome spontaneous fermentation routes. Now, for his master's studies, John Lubale, one of my master's students, studied the effect of these 3 methods of souring on phenolics and antioxidant properties of finger millet gruels. And as you would see here, souring by the use of Lactobacillus plantarum starter culture, or by using a backslopped inoculum, or by exogenous acidification with lactic acid, enhanced extractability of flavonoid compounds and also enhanced radical scavenging properties of the gruel. So you'd see here, this graph is showing you what we got in terms of the total flavonoids for a red finger millet, and you will see if you look at the blue bars, I've put a line on top of them to show you the increasing trend as you go from the unsoured flour to the fermented flour and also to the spontaneously fermented flour. You find the same trend for the red-brown finger millet, and you find a similar trend in terms of the antioxidant properties for the red finger millet and the brown finger millet as well. So this gives you an idea of some of the work we've done at utilization level 1 to show the role that phenolic compounds can play in promoting health in foods. We'll now move on to level 2, where we're looking at modern high-level technology processing methods. And this, I'm going to talk about condensed tannins a lot here. And I will be talking about tannin sorghum instant porridge prepared using extrusion cooking. And this was work done by my PhD student, Dr. Martin Adakwayado. Now, I need to give you some background here. Some sorghums contain condensed tannins, and the sorghums that contain condensed tannins are classified into 2 main groups, type 2 and type 3. And the difference lies mainly in the mode of deposition of these tannins, and I'll speak about that a bit later. The condensed tannins offer an agronomic advantage because they protect the tannin sorghums from being eaten by birds in the field. However, the condensed tannin sorghums have relatively limited utilization compared to condensed tannin-free sorghums. Why is that? It is because the condensed tannins have adverse sensory effects. They exhibit or they provide bitterness and astringency, and they also have adverse nutritional effects. They can reduce digestibility of proteins and starch. They can inhibit digestive enzymes. Now, in tannin-type sorghums, the condensed tannins are actually located in the outermost layers of the grain, mainly in the testa and the pericarp. Therefore, decortication is actually a simple method by which condensed tannins can be removed or reduced. And this was actually demonstrated by one of my master students. So working under the supervision of myself, Professor John Taylor, Professor Reddy Koch, my master student, Mr. Andy Lemgluli, showed that condensed tannin sorghum could actually be decorticated to a particular extraction rate such that it begins to actually resemble a non-tannin sorghum in terms of its sensory properties. Now, for Martin's work, we decided to use a different approach where we decided to prepare an instant cereal-based porridge using extrusion cooking. Instant porridges are consumed extensively by urban dwellers. So we decided to use extrusion cooking to provide or produce an instant porridge, but then use the whole grain tannin sorghum. And we're interested in three main questions. Number one, can extrusion cooking be used to break down proanthocyanidins in tannin sorghums? Secondly, will type 2 and type 3 tannin sorghums show differences in the susceptibility of their proanthocyanidins to break down? And then thirdly, how bioaccessible will the proanthocyanidin species from type 2 and type 3 tannin sorghums be? So that's what we did. And this is what we found. We found that for the 2 tannin sorghum types, extrusion cooking reduced the proportion of oligomers and actually increased the proportion of mainly dimers. So this is what we were succeeding in doing. We were breaking down the polymers and oligomers and forming mainly dimers and monomers. What about bioaccessibility? We found that whilst the bioaccessible fraction of the type 2 tannin sorghum instant porridge contained mainly monomeric and dimeric proanthocyanidin species, that of the type 3 tannin sorghum contained monomeric all the way through to hexameric proanthocyanidin species. And this is a clear indication of differences in extractability of proanthocyanidins in the 2 tannin sorghum types. And this model just shows you what we're trying to say here. Proanthocyanidins in type 2 tannin sorghums are deposited in vesicles, which are in turn attached to the cell walls of the testa. On the other hand, proanthocyanidins in type 3 tannin sorghums are deposited freely along the cell walls of the testa. So you can imagine a situation where in order to release and break down proanthocyanidins in type 2 tannin sorghums, whatever processing method that is being used, in this instance extrusion cooking, it needs to break down the vesicles before the proanthocyanidins can be released, which is not the case for type 3 tannin sorghums. Therefore, this makes the proanthocyanidins from type 3 tannin sorghums more extractable and potentially more bioaccessible than from the type 2 tannin sorghums. And in agreement with these observations, we went on to look at cellular antioxidant activity as another way of determining health-promoting properties, and in agreement with the observations from the bioaccessibility studies, the porridges and digests from the type 3 tannin sorghums, they showed greater protection of KECO2 cells against oxidative damage compared to type 2 tannin sorghums. So we'll move on to utilization level 3, where I'm now going to talk about the marama bean, which maybe some of you may not have heard of before. And I'm going to talk about work done by my PhD student, Dr. Jeremiah Shilembe, where he was looking at marama bean seed coats as sources of bioactive phenolics. Now, the marama bean is actually an underutilized legume crop. It is native to the Kalahari Desert, found growing in the wild in southern parts of Africa, mainly in South Africa, Namibia, and Botswana. Now, usually the marama bean is dehulled, and the cotyledons are consumed mainly as a roasted snack, and the seed coats are discarded as waste. Part of Jeremiah's work was to conduct an in-depth characterization of proanthocyanidins in the seed coat of marama bean. So he prepared water extracts, acidified water extracts, did phenolic characterization, and determined bioactive properties as well. So what he found was that the aqueous extracts from the marama bean seed coats contained extremely high levels of condensed tannins. You can see from that table, the levels you are getting for the marama bean seed coat way higher than for the sorghum bran. And then the extracts from marama bean seed coat also exhibited significantly higher protein precipitation capacity compared to aqueous extracts from condensed tannin sorghum bran. So this told us that definitely we have condensed tannins in these marama bean seed coats. So we went on to try and characterize the condensed tannins. So the aqueous extracts were separated into oligomer and polymer fractions, and the constituent proanthocyanidins were subjected to thiolytic degradation using benzyl mercaptan, which is a nucleophilic agent. And what happens is that the benzyl mercaptan tags the extension units of the proanthocyanidins and converts them to what we call benzyl thioether derivatives. But then it leaves the terminal units free and untagged. So after you've conducted a thiolytic reaction, you conduct an LCMS analysis of the mixture you've got, and you'll be able to identify the extension units and the terminal units. So that's exactly what we did. And what we found was that the terminal units of proanthocyanidins from marama bean seed coats consisted of, and I'm going to spell out some names here, epigallocatechin 3O gallate and epicatechin 3O gallate compared to just catechin in the proanthocyanidin from tannin sorghum bran. What about the extension units? The extension units from proanthocyanidins from marama bean seed coats consisted of epigallocatechin, epigallocatechin 3O gallate, epicatechin, and epicatechin 3O gallate. In contrast, the extension units in proanthocyanidins from condensed tannin sorghum bran consisted only of epicatechin. And so from these results we could actually propose a possible structure for a B-type proanthocyanidin in marama bean seed coat, where the structure consists of those extension units, epigallocatechin, epigallocatechin 3O gallate, epicatechin 3O gallate, and epicatechin, and then epicatechin 3O gallate and epicatechin 3O gallate as the terminal units. And you compare that to the sorghum tannin structure, and you see how relatively simple that is compared to the marama bean tannin structure. So we found that the structure of the proanthocyanidins from marama bean seed coats was quite unique compared to other legumes because of the high percentage of galloylated units. And then also the marama bean proanthocyanidins had higher mean degree of polymerization compared to tannin sorghum proanthocyanidins. What about health? We found that these aqueous extracts showed protection against human red blood cell hemolysis and LDL oxidation. So I hope I've been able to give you an idea of how important these African grains are in terms of their phenolics and their ability to promote health. And the question then is, what next? And this is quite important. And I would like to offer three things that I think we should be looking at. So, like I said, the research data I have presented to you shows the potential health promoting properties of these African grains in relation to their phenolics. So I'll now briefly mention these three areas which I believe we should be having a closer look at, and which will be an area of focus when we come to African grains and their health promoting properties. We would like to expand our research into phenolics in African grains and their health promoting properties to now begin to look at in vivo work using animal models and human intervention studies. African grains and their foods, they are generally under-researched in this regard. And this will provide some really good insights into things like bioavailability of the phenolics and their metabolites and how this impacts health-promoting properties. Now, these African grains and their foods are quite intricate and complex systems, and they contain many other components apart from phenolic compounds. So what I'm showing here, that is actually a wheat grain and not part of the African grains that I'm talking about. But I think what you see there also applies to African grains, because apart from phenolics, they contain all these other substances as well. So these phenolic compounds do not work in isolation. So we believe there is a need to adopt a more holistic approach and study how phenolic compounds work in concert with other bioactive compounds of the grain to promote health in a fashion similar to how a music orchestra functions. And now thirdly, I believe that our research should make a difference in the lives of consumers. It's wonderful to publish great papers, but I believe we should make a difference in the life of consumers. And the formation of strong transdisciplinary research consortia would put us in a strong position to achieve this. And these are opportunities we will be looking out for. It is easy to see how research on health promoting properties of African grains and their foods can have a positive effect on the health of the general population. However, I believe there are opportunities to improve the livelihoods of consumers as well. Professor Bruce Hammaker is a collaborator of mine. He is distinguished professor of food science at Purdue University in the US. He is a strong advocate of the hub and spoke food innovation model. So one can envisage such a model being implemented for an African grain-based food with proven health benefits in rural, urban, and semi-urban communities. And this would promote entrepreneurship, it will build capacity, and empower important people groups such as women and the youth, and also promote health. So there is no doubt that African grains and their foods will play an ever-increasing role and be critical for health and well-being, especially in Africa. And we believe that their promotion and increased utilization will put us firmly on a path towards helping to achieve the sustainable development goals of no poverty, zero hunger, and good health and well-being. It is my hope that I have succeeded in raising awareness about African grains and foods, and that at the very least, I have put a pebble in your shoe in this regard. I'd like to acknowledge a few people. First of all, starting off with my family, I'd like to acknowledge my late parents over there. I'd like to acknowledge my better half, who is actually watching this from Memphis, Tennessee. And one thing about her is that she has this habit of asking very probing questions about food. And what that does for me is that it gets me thinking, because it actually gives you the perspective of food from a non-food scientist point of view, and that is also quite helpful. I'd like to acknowledge my Uncle Fred and my Aunt Alice in East London, acknowledge my brother and his family in Ghana, Yao, and my brother Ibu and his family in the UK, and also acknowledge my cousins, and there are so many of them. I'd like to say, please forgive me if I don't have your picture here. I couldn't get a lot of pictures. Over the years, I've had the privilege and the honor of working with so many students, so many masters and doctoral students, and I've listed some of them here. And they've played a very, very important role in contributing to this research area. I would also like to acknowledge high school mates of mine who are watching now. I'd like to acknowledge two groups in particular. OAA 85, they know who they are, and my fellow mates from boarding school in Livingston House. I'd like to acknowledge all of you. And then I'll also like to acknowledge my colleagues in the department. We started off small as the Department of Food Science, and then we increased when we became the Department of Consumer and Food Sciences. I would like to acknowledge all of them. And then I'd also like to acknowledge two very important and special people, Professor John Taylor and the late Professor Amanda Minna. And what I have got from these two, which is priceless, is good and excellent mentorship. And I would really like to acknowledge them. They always offered their brains to pick, they offered their ear to listen, and definitely they always pushed me in the right direction. Thank you very much indeed.
In particular, I'm heartened to see that his postgraduate students are deeply embedded in this process and that they contribute actively in pushing the boundaries of knowledge. Getting to this point, and also the inaugural address this evening, which is a high point in any academic career, takes a lot of support from many different people, not just career-wise, but also to the event this evening. In particular, I'd like to a word of thanks to the Vice-Chancellor and Principal, Professor Coupe, for being here and spending time with us. Particular word of thanks also to Professor Elna Buys for joining us this evening. And then Professor Duodu, I'm acutely aware that getting to this point and having achievements at this level does not come without cost to your personal life. And I want to extend a particular word of thank you to your family and your extended support systems at home to allow you to have come this far. Thank you very much. Finally, I also want to extend a deep word of thank you to you personally. I know that you play a very active role in the faculty, and you're not shy to step up into leadership positions when the time requires. And in particular, thank you very much for being the acting Deputy Dean for some time. You really made a contribution, and you really helped me to keep the ship steady during the pandemic. Thank you very much for that. Thank you for Spatial Studios, for professional service as ever. And I'll be remiss if I don't mention Marie Venter and the rest of the marketing communications team of the Faculty of Natural and Agricultural Sciences for pulling this event together. Friends and colleagues, thank you very much for joining us. Stay safe and vaccinate.